Hey, this is Matt Schoenled. In this video, I'm gonna talk about signal amplification. When we think of it, especially for source differentiation, the fluorescence microscopy usually be our first choice. But sometimes it causes some problem because of wavelength overlapping. In 2016, a research paper published is about a new way to amplify the signal for cancer cell detection called Signal and Emission Reactive Release Amplification, or SIRA. But what exactly it is, I'll explain its concept in 3 different parts. How to amplify, the labels, and the detector. So the first part, they use the MAC particles to connect much more little particles that have been already labeled. And then the second part, they choose the short peptide chains as the labels so they can design their own amino acid sequences. So it also means each kind of labels will have their own molecular weights. And the final part, they use the mass spec system as their detector. And because of its high sensitivity, the short peptide chain can be easily differentiated. So that's it for today's 2 minutes learn and share. If you'd like to know more information, please check our description below. And if you like it, please subscribe for more videos.